So much, and really thank you for having me. I'm pretty excited to talk to everyone today about this application that I've developed here while at Slack. So yeah, and I mean, the introduction said that, but I'm from the University of Pennsylvania. I work with Dr. Eric Detzi, and these are some of my coworkers. I mean, I'm one of the first students in Dr. Detzi's group, and so it was really great to build up a new lab and start doing experimental science in the group. And following some of the work that I did there, I was awarded this DOE SCGSR fellowship, and I've been working for Dr. Nelson Wecker's group at Slack, which has been really awesome. And I learned a ton about this particular technique. But some of the processing that I would have to do for my experiments, I really wanted to make this workflow much simpler, especially for someone who's an experimental scientist like me, where using a text-based tool might not be as user-friendly to do this type of analysis. And so what I found is that, when you move to Palo Alto, the home of Google and Facebook and a lot of software startups, is that you really have to build an application while you're here. The difference here is that I'm not pitching you all for money from some venture capital firm. I'm basically trying to help implement this type of program at various national labs, and today at APS. So here is the basic pitch behind that. So with TomoPy, which is a really well-known application for tomography data processing developed at APS, the basic premise is that you write a script, you edit that script for each data set, you run the script, and then you import whatever result you get from that script into some image processing software, some image visualization software. And then you look at the data and you see, okay, did this work? Did the alignment work for my tomography data set? Did the reconstruction come out good? And you have to ask yourself, am I satisfied with this result? If you are satisfied with the result, then you take that data set and you pull it into another software, Dragonfly or a Viso, to do some further analysis on this reconstructed 3D image. But if you're not satisfied with the result, you have to think about what you did with the previous settings in your script. And then you have to make some alterations to that script. And then this process continues over and over and over again. And so for the average user of tomography, they come in and they have to learn this new Python API to figure out which of these variables are going to impact the final reconstruction. And so this is complicated for the user like me who doesn't really know anything about tomography data processing. So what I've built is TomoPyUI, just a user interface for TomoPy. And what it can do is you specify some data path, and it will import and normalize the data automatically. And then there are various different steps throughout this processing protocol where you can interact with your data. You can visualize it. You can find the center of rotation automatically and interactively. You can choose these various options, and then you can run the analysis. So you run this. It's basically like it creates a script for you, and it runs. Then you can visualize the results of whatever you're doing, either alignment or reconstruction, immediately after you run this program. Then if you're satisfied with that, then you can take it to segmentation. But if you're not, then you can look at your previous settings for all of the different options that you chose and the center of rotation and all of these things in metadata. And that metadata I've created, and I'll show you here in a minute, you can see those settings. So if you specify this new path, you can see what you've done in the past. And so this process speeds up the processing timeline significantly. So I'm going to go through each of these steps in this presentation. So yeah, so this is basically what you'll see. It's all housed in Jupyter. And so all you have to do is run this cell after you install the program, and it will create this dashboard. And the dashboard contains all of these different tabs so that you can import your data and visualize it immediately. So what you're seeing here is I just pasted in a tab, pasted in a file path, and it recognized that there are several different energies associated with this particular acquisition. So you can then choose those energies and upload your data. Now this takes a little bit. And so, it'll show you the progress of uploading the data into memory. And then eventually, after it does the normalization, it will pop up into this image visualizer here. So with the visualization, just to show you how you can interact generally throughout the entire application, here is this new uploaded thing into the memory. So you can slide through just like ImageJ. You can change the color scheme. There's a histogram over to the right of the image visualizer. And then what's kind of a critical thing behind some of the general processing here is binning or downsampling of the data. So you can easily switch between various downsamplings to slide through these images in a faster way. There is this problem with Jupyter notebooks where the widgets, there's a relay between the front end and the Python kernel that causes lag sometimes when you're sliding through this type of data. And so if you wanted to really quickly slide through this data, that's going to be key, because you don't want to have lag when you're trying to find the center of rotation, which I'll show you in a minute. There's also other tools to look at, quickly view the sinograms of the data. And as you can see, this data isn't really well aligned at all. There's a lot of jumps in the sinogram space. So moving forward, I've also created a pre-processing tab where this is in case you wanted to apply some kind of filters to your data. So for example, what I'm going to show you here is that you can add pre-processing methods, and maybe you want to add a median filter or a Gaussian filter, and this will show up after you add them into this methods list, which you can then run, and it will show up on this side after pre-processing. It'll save the data, and then you can continue on with your alignment or reconstruction of your data. So there is, in this tomography processing pipeline, finding the center of rotation is actually critical to having a good reconstruction or a good alignment. And I've built tools on top of this visualization, this visualizer, so that you can quickly do this. Typically, you have to, if you're doing this with a text-based tool, you would run one of these particular programs inside of TomoPy, and then it would export all of these images into a folder, which you would then pull up in either like Windows Image Viewer or ImageJ, and you would see which center of rotation is the best center of rotation for your data. But with this tool, you can see the center of rotation using this red line. You just visualize that. Then you can slide up and down on this axis to determine which slice of the data that you're going to reconstruct. And so that's what you're seeing here. I'm picking this particle to find the center of rotation in. And then I click this button and it will find, it'll reconstruct this slice of data along at various centers of rotation. So now I can slide through these centers of rotation to see which center of rotation provides the best reconstruction, and then I can narrow that search range down. So now I've gotten pretty close to the center of rotation, but I want to really hone in on the actual center of rotation with a smaller search range. Then if your data has some kind of tilt in it, so this is an instrumental thing. If the axis of rotation isn't perfectly perpendicular to the ground, then what you'll come up with is an axis that is off alignment. So I've built in a feature here to add multiple centers of rotation. And so this is what you'll see here. I'll reconstruct this again, and it's going to be clear to you that this is not as good of a center of rotation as this first point that I showed. So again, I'll sort of hone in on that. And then you can see that this is a tilted axis. And again, this is critical for you to be able to reconstruct your data and get a high quality reconstruction throughout the entire y-axis of your data set. Okay, so after you find the center of rotation, all of that information is filled in throughout the rest of the application. And so what I'll show you next, which is a critical thing in nanotomography, is data alignment. This is not as big of a problem in micro CT, but using this alignment feature that's built into TomoPy, I'm able to automatically align my data in a much quicker way than I would by hand aligning with another program like TXM Wizard. And so this is this alignment protocol here. So what you're seeing on the left is just the raw data, the imported projections. But in alignment, especially using automatic alignment, there are many features within the data set that you might not want to include in the alignment. The basic way that the alignment works is that you first reconstruct using these projections, then you reproject the, it's basically like simulating projection images, simulating the raw data, using that reconstruction. And then you compare the original data set and these simulated reprojected images. Then you'll take that, the comparison using phase correlation, and you'll shift the original data set and iteratively do this to get a much more high quality reconstruction and hopefully a smooth data set. So what is good about this particular way of doing this within TomoPyUI is that I can crop the data set to what I want to reconstruct. This not only makes this a faster process for the actual reconstruction and reprojection and phase correlation matching, it is important for the actual automatic alignment because you might not want to include part of that data set in this. And so what I'll show you here is that I'll set some settings up and add padding and all of this other stuff. And I'll also downsample the data. So again, all of these things, all of these small steps, they take a lot of time. And if you're using a full resolution data set, it's going to take a really immense amount of time to do this alignment. And by the end of that process, you don't even know if you have done this correctly. And the alignment can come out really poorly. And so then you kind of give up on doing the automatic alignment. But if you're able to interact like this with the data set, it's much quicker. And so what you'll see here is I'm downsampling this by a factor of 8. And I've built some wrappers around what's called Astra Toolbox, which is a really kind of sped up version of a lot of these reconstruction algorithms. I've built a wrapper using CuPy to use the GPU effectively. And so you'll see this in a second, that the alignment process is really quick. And yeah, so I'll go ahead and start the alignment. One thing that's nice is that you can see at least one projection image, how this original data set compares to a reconstructed data set. So you're seeing here that there's a lot of, let me go back a little bit. So again, this alignment algorithm works by taking the original data set, reconstructing it, reprojecting it, as you can see here, and then matching these two images. So matching this image, and then shifting this data set to better match this image. So you can see in the shifts of all of the different projection images in X and in Y down here, and that can kind of tell you what's going well with the alignment or what's going really poorly. You can see that in this shift in X and Y graph here along this alignment process. You can also see how well this is converging. And so it'll go through these iterations, and then finally it'll just immediately show up with your original data set, and then the full data set shifted to those shifts that you found. So you can kind of see this here. You can link both of these up, and then you can see how well this alignment really worked. So it's much more jittery here than it is on this side. Right, so you can see this particle jumping around, and that's because I've done the automatic alignment, and it's come up with some better solution for the projection images. So then you can click this button, and it'll again downsample your data, and it will update the plots throughout the application. So then you can either do another alignment, or you can do some reconstruction from this now aligned data set. And then you could take it to your Viso or Dragonfly to do segmentation and all this further analysis that you do with the reconstructed data set. So this is, good in theory, but does it really work in practice? And so I have a couple data sets that I'll show you that are real. So this is this problem with nanotomography. Again, I might not have explained that for people that aren't familiar with this. There's this huge problem in nanotomography caused by the run out of the motors. So basically, when you're turning the motor to collect a projection image, you turn the motor, and then you collect this projection. You turn it again, you collect another projection, but there's imperfections in the stage, and so what happens is the sample is jumping around in the field of view. You can clearly see that this data prior to alignment is no good, and what you would end up with is a super blurry reconstruction. So using automatic alignment, you can or manual alignment, you can sort of fix this problem by trying to make this as still as possible as it's rotating through the data. And so now what I'm going to show you is alignment within this area. I don't use the full data set. This is a high resolution data set. It's a 2048 by 2048 and 360 images. So what I'm doing is I'm cropping all of that out, and then I'm going to compare a downsampled alignment and reconstruction and a full resolution reconstruction within this area. And so using the tools in TomoPyUI, you can see that the shift in X pixels and the shift in Y pixels between the full resolution data set and the downsampled by 8 resolution data set are very similar. So basically, downsampling in alignment is nearly identical to just full alignment with the full data set. And so here in blue, again, these are the shifts that you're doing to the original projection images in X, and then in Y. And the black line here is the difference between those shifts between the full resolution data set and the downsampled data set. So you can clearly see that there's barely any difference in the X, and there are some differences in the Y, but if you look at the scale here, the amount of pixels that it's shifting in Y are not nearly as large as in X. So, I mean, one thing you could do is you could quickly align with a downsampled data set, and then to really hone in on the full resolution data set, you could do a few iterations using the full resolution. So this is what this data looks like after alignment. So you can see that that clearly fixed the problem there. Yeah, and so this is one more data set. And so this is a comparison between raw data. You can see this sinogram again. This is actually one of the data sets that I was showing previously when I was importing and showing you visualization. This is a sinogram on this particular particle, and it's really jittery throughout this entire raw data set. The sinogram shape is changed significantly when you use the protocol in another software called TXM Wizard that Dr. Eugene Yu created here at SSRL a while ago. And so what happens with the TXM Wizard is you're clicking on this particle and you're putting it arbitrarily in the center of the data set. And so this changes the shape of the sinogram significantly. But what you'll also notice is that the jitter is still there for one iteration of this manual alignment. You have to click through 180 images in order to manually align your data set. Now, after you do the automatic alignment from the raw data set using the TomoPyUI and TomoPy's automatic alignment and reconstruction algorithm, you can clearly see that the resolution of this reconstruction is much better than either the raw or the manually aligned data set. And it's the difference here between the automatically aligned data set and the manual and then automatically aligned data set is that the resolutions are pretty comparable, but actually the shape of the final particle has kind of changed a little bit. And that is because of this this large change in the shape of the sinogram. So you can see that this comes to like a much sharper point than this does, and the automatically aligned, the shape, at least the perimeter of this particle, looks much more similar to the raw data set that we have. So that sort of brings me to how this whole structure of this code is arranged. And basically, I have various different parts of the application. I have the front end, which is housed in this widgets folder. And so there's all of these different submodules that are connected to all of the different processes. So importing center of rotation finding, the analysis, which is the reconstruction and alignment algorithms here. Well, so the front end of those tabs. And then all of these are connected through this plot submodule. And so the image visualizers that you see throughout the application are just a class that's in this plot submodule. So it's kind of this modular thing where I can, take that visualizer, I can add widgets to it depending on what I want. I could add, the center of rotation lines and all of these different sliders just for a specific part of the application. And so all of the front end obviously talks to things that are going on in the back end. And for you at APS, the big thing is, in order to get your data into the software, is to change this IO. And I've been talking to multiple people about how much development is really just about IO compared to the cool stuff that you can do with image visualization or visualization in general. And I mean, it's really like 50% or more of the work is in this IO, where everyone stores their data in a different way. Each beamline has a very specific data metadata storage structure. And so I've made this a sort of, I've made a base class, and I've made a class hierarchy in order to create sort of a template for people to use to get their data inside of the software. And so Dr. Francesco De Carlo has been working with me on creating one for APS. And so, we have now a few different beamlines that have written code for this, or I've helped them write code for this. And we're trying to get more and more people involved in bringing their data into this application. And really, I mean, you can take your data after you do the alignment and reconstruction in my application, and then apply it to, some kind of bulk script for your whole data set. If you wanted to take this data and align one thing, and then align one data set, and you could use that you could use all of the metadata that's stored throughout this whole process on all of your data. And so, yeah, so this is the general idea behind TomoPyUI. I know this is been sort of a 30-minute conversation, but I'd like to have some discussion about how this could be used or implemented at APS, and what you guys, any feedback that you could give me would be really awesome. It's the code is entirely open source, and I'll send links through the chat here in a minute. If you can check out the open source code, we have a website, all of that information. There's tutorials on the website on how to install the software and how to use the software in general. So with that, thank you so much. Thank you all so much for having me. I'm really excited about this type of work. So please don't hesitate to ask me questions or send me any feedback or anything. So thank you so much.
Great, wonderful talk, Sam. If we all could, maybe we could unmute and give Sam a round of applause. Right. Wonderful, thanks again. So yeah, I'd like to open up now for questions for Sam. If anyone has a question, if you want to, you could either put it in chat, or just you could raise your hand or unmute and start talking. So I'll ask you one, Sam, to get started. Is what does it take to actually, what platform are you running this on in terms of, is it running at the beamline? What kind of hardware would you need to run this? Could you elaborate a little bit more on like the actual installation process?
Yeah, so a lot of the features that are in the application require CUDA. And so you would at minimum need a GPU with a that's capable of CUDA, I think 10.2, CuPy 10.2 or higher. So that, and again, I have pretty detailed installation instructions on the website, but installing CUDA is like always kind of, a pain, and especially for people that don't know anything about installing that kind of software. But it's a kind of simple installation process, and I've gotten people up and running at UCLA just on a kind of home computer. At SSRL, we have a sort of dedicated workstation, or a few that are that can run it. But because it's built in Jupyter, there's possibilities of using it on Jupyter Hub. And Jupyter Hub, if you're not familiar with this, is a server-based Jupyter. And so there's the potential of, and it would just take more work. There's a potential of just making these like a Jupyter server basically, and people could just log into this, and remotely do this kind of work on, much faster computers than just a desktop or a regular workstation. Those are all possibilities, but yeah, it'll just, take some more building of it. So, I mean, does that answer kind of your question there?
Yes, yeah. I guess kind of to follow up on it, do you have plans at SSRL, or does SSRL have plans to kind of set something up on a larger Jupyter-based infrastructure that you know, or what's
not that I'm aware of. We don't have those plans in particular. I mean, again, this project kind of developed organically, where I was just trying to make this more of a user-friendly process for myself and for my data processing. And so my DOE SCGSR is actually mainly focused on the experimental side of things, which I'm also, I have beam time in a couple weeks. So it's really just, after my contract ends in September, I don't think there's any plans to further scale up on this kind of server thing. But I have talked to people at ALS, one of Dula's people who work for Dr. Dula Parkinson there, about this possibility.
Okay, great, thank you. Yeah, this is really wonderful work. Let's see, any other questions, comments from the audience? Victor.
Can you hear me?
Yeah.
Yeah, so yeah, I'm Victor. I'm again from the imaging group here at APS, working in Francesco, and I also work quite some time with the TXM data and also with this alignment problem.
Mm-hmm.
Yeah, I just want to ask, have you thought about implementing some other alignment algorithms? Because, for instance, in our case, we are doing fly scan, continuous scan.
Right.
And we don't have this jitter problem. So in our case, the sample just moving in one direction.
Right.
And when I tried one of these algorithms with alignment with the projection method, so typically they don't work in 90% of cases.
Yeah.
So some other more advanced algorithm can be.
Yeah, and I know that there you all have developed some newer algorithm. I was basically, so again, I saw this application TomoPy, and it comes with all of various different stuff. There's pre-processing, center of rotation finding, automatic center of rotation finding, which I've implemented here, and there is a couple of alignment algorithms kind of based around the same function, where you're doing the projection and then reprojection and comparison using phase correlation. I know that there are more advanced algorithms that can really help out with this, but they're not just available on TomoPy. So implementing those, again, it's totally possible. And the way I've set up the code through this class hierarchy, you would be able to add that type of thing with a couple clicks to the software. Like, say I want to have a little radio button that says use this algorithm or this algorithm. That would totally be something that you could definitely do. And again, it's just that I had access to this and not enough time to implement full other types of algorithms. So yeah.
I will be happy to contribute.
Sorry, I didn't hear you.
Yeah, I will be happy to contribute and probably add some method to your package.
Yeah, that's great. I mean, it's open source again, and yeah, I guess before I forget, I should probably just send the links to the application. So I'll just send them through the chat right now. But yeah, it's open source. Some people have contributed. A lot of people have reached out, and yeah, it's open for anybody to submit a pull request. And I can, if you're interested in doing one thing in particular, like writing an import class, I can help you along the way. It's because I mean, I know the code because I wrote it. So it's, it helps to have a little explainer.
Hi, Sam. This is Junjin from X-ray microscopy group. And in your slide, you mentioned the TL rotation axis. So your code can assign like different rotation center for each slice of the sonogram. Is my understanding
Yes.
correct?
Yes.
And so after reconstruction, each slide is like based on the rotation center and then assembled to a 3D.
Yeah, so maybe I can go back to that tab. I stopped sharing, so it'll take me 1 second.
Okay.
Okay, this is the tab.
Yes.
So yeah, so basically the way that reconstruction, for everyone who's maybe not familiar with it, is you're taking a slice through all of these projection images at this particular value, so of y, and you're using that sinogram through that for that slice to reconstruct one part of your reconstruction domain. And so what you are doing here, you have to use a center of rotation. You can use just one center of rotation, or if your acquisition instrument has some physical tilt axis, then you would want to reconstruct along multiple centers of rotation throughout that y-axis. And so you can see this at the end, that the best the best one that I came up with for this particular data set on this instrument, you can see that it's a tilted axis, right?
Yeah.
So it selects these two different points, and it just does a linear fit between those 2 points. Basically just makes a line.
Okay, great.
Yeah.
Second question is about your slide of, the automatic alignment. And also the other example is manual automatic alignment.
Yes.
And so in practice, how can we evaluate like which one is better?
Yeah, I mean, it's a tricky question because we don't necessarily have like a ground truth to this. Now, I guess I think in the ideal case, you would have a ground truth. For this data set, I don't have a ground truth measurement. You could, the issue, so let me back up. The issue with the automatic alignment in general is that things will fly in and out of the field of view for this data set. And for the parts that are flying in and out of the field of view throughout the data set, it will falsely align to those features. And what you may end up with, and what you can see in the aligned data set sometimes, is that there's like a big lateral shift in the full data set. And if you look at the sinograms in those cases, there are these, it doesn't look like a normal feature in the sinogram space. So it's a difficult problem to say, okay, I know that this is better, but evaluating it just based on the resolution before and after alignment is just one way you can do that. Ideally, you do have some kind of ground truth that you can say, hey, this is actually better and more true than what the manual alignment does. Do you see, I guess? So
here in the alignment, do you just apply only translation alignment and no other, for example, affine transformation? Because if we look at the sinogram on the top and the bottom, it seems the sinogram's width, the signal width is different. So here is just, the kind of scale bar issue.
Yeah, this is a scale bar issue for this particular data set. But so what you're doing with the manual alignment in TXM Wizard is you'll have this full, let me just go back to a slide with all of these kind of data sets. So what you're doing with TXM Wizard is you're taking each projection image. So this is before alignment again, so you can see it's really jittery. What you would do is you would take the top point of this and you would click that, and then it would move to the next projection image after shifting that projection image where you clicked to the very center of this, the center pixel here. So it would shift the entire data set to that center pixel. And then you would end up with something that doesn't rotate necessarily around the center of rotation, but it rotates directly on the center pixel. And you're setting the center pixel to be the center of rotation. So what that ends up doing is it changes this shape of the sinogram. It's still just a translational thing. There's no affine stuff going on. This is just you can see that the center here has been shifted so that it's rotating just around that center pixel. I mean, I don't know if that was clear, but does it kind of answer like how those two different things work?
Yeah, great. Thank you so much for answering.
Yeah.
All right, that's a great question, Jinjing. Let's see, do we have any other questions for our speaker? Doesn't sound like it, but I'm sure that Sam would be very happy to field other questions if you think of things later. I'm sure Sam would be happy if you reached out via email or some other mechanism, and you can have a chat that way too.
Yeah, and you can post on the GitHub if you want. If you have any suggestions or issues, you just raise an issue. Yeah, by the way, I just put the GitHub on the chat, and the read the docs is right above that. So you can scroll through those, and if you have any suggestions, don't hesitate to throw something on. It's totally open source, like I said, so.
Okay, wonderful. So Sam, thank you again for the great talk. This was really informative, and I think gives people at the APS a lot of great ideas on how to proceed in this space. So thank you again.
Thank you so much for having me. It was really a pleasure to meet everyone.